Hi, my name is Usman Muazzam and I am a research intern at Fred Hutchinson Cancer Research Center. My research project is titled Providing Proof of Concept for the Application of CRISPR-Cas9 Genome Editing and the Reactivation of Fetal Hemoglobin to Treat Beta Thalassemia, which is otherwise known as sickle cell anemia. Sickle cell anemia is a very common monoallelic disorder that falls under the umbrella of beta hemoglobinopathies. Sickle cell is caused by a defect in the beta chain in adult hemoglobin. This defect causes red blood cells to take a sickle-like shape, giving them a lower affinity for oxygen and the tendency to clump together in blood vessels, which causes anemia and potential blood vessel blockage in patients. It has been observed in the medical world that sickle cell patients who happen to have HPFH, which is hereditary persistence of fetal hemoglobin, have much less extreme symptoms. These patients have continued to produce fetal hemoglobin into adulthood, along with adult hemoglobin. Because fetal hemoglobin does not have a beta chain, the defect associated with sickle cell is present in far fewer of the patient's red blood cells. Therefore, individuals with HPFH have their defective hemoglobin essentially replaced with functional fetal hemoglobin, making the symptoms of sickle cell anemia far less effective and extreme. Theoretically, if we could reactivate the fetal hemoglobin gene to allow patients to produce fetal hemoglobin, we could replicate the effects of HPFH and alleviate the major effects of sickle cell symptoms for patients. Now, in order to accomplish this goal, we will be applying a stem cell-mediated therapy to hematopoietic stem cells using technology to destroy the fetal hemoglobin repressor, thereby reactivating fetal hemoglobin production. We will be using nanoparticles to deliver this treatment to the stem cells. The anatomy of the nanoparticles are built around a collection of polymer and mRNA, surrounded by a coating and targeting ligands in order to bring the therapy into the cells. Inside the nanoparticles, we will be putting Cas mRNA and protein. The Cas9 technology will make a precise cut at the repressor site of the fetal hemoglobin gene, which will reactivate fetal hemoglobin production. By carrying out this process in the hematopoietic stem cells, this genetic edit will be carried down to the rest of the blood cells, including red blood cells, allowing the red blood cells to produce and carry fetal hemoglobin. To start, we will be applying this treatment ex vivo to human CD34 positive cells. This includes a proof of concept step to illustrate that the nanoparticles are being taken up by the cells, and a proof of application step to attempt to create the desired cut at the fetal hemoglobin gene repressor. The first phase of the project revolves around the nanoparticles themselves, which entails constructing them with a variety of titrate ratios and seeing if they are taken up by the cells. We start this process by thawing some CD34 positive cells on day 1 and then incubating them. The next day, we construct our nanobodies to the specifications of the ratios and transfect our cells, letting them incubate overnight once again. The nanoparticles themselves consist of GFP mRNA, Cas9 mRNA or protein, and guide RNA. The GFP mRNA is there for confirmation by flow cytometry that the nanoparticles were taken up successfully by the cells. The Cas9 and guide RNA serve the purpose of facilitating the cut at the repressor site. On day 3, we harvest our transfected cells and analyze them with flow cytometry, with adequate GFP resonance affirming that the nanoparticles were taken up by the cells. When using Cas mRNA, we use an untreated cell sample and GFP-treated cell sample as our controls. For the nanoparticle-treated sample, we varied the Cas9 guide ratio between each of the samples in order to test which titrate ratio maximized the efficiency of the nanoparticles. According to the results of the flow cytometry, it appears that in each sample, the cells were successfully transfected with the nanoparticles. When using Cas9 protein, we use similar treatment for the control samples with an untreated sample and GFP-treated sample. For the nanoparticle-treated sample, we tested 2 Cas9 to guide ratios with 3 samples each, with each sample having a different cell count. In this version, we tested both titrate ratio and cell number, observing which combination maximized efficiency. We received similar results to the mRNA experiments, observing that in each sample, the cells were successfully transfected with the nanoparticles. Unfortunately, we were unable to complete enough repetitions of the experiments to definitively determine which combination of ratios had the best efficiency. Having observed that the nanoparticles were taken up by the cells, our next step was to see if the precise cut at the fetal hemoglobin gene site had been made. In order to do this, we extract the DNA from our cell samples and amplified a portion of the fetal hemoglobin gene repressor sequence using PCR. We then analyze the sequences using TIDE software, looking for a precise and clean cut at the desired location. Here we have the data from our TIDE sequencing. Unfortunately, many of our samples appear to have no sequence at all. This points to some sort of error with the actual sequencing of the samples. However, we received some promising results from the samples that were sequenced correctly. From each batch of experiments, we received a sequence with a cut at the desired place in the sequence, shown by the high efficiency percentages and correlation values in sample 3 of the RNA samples and sample 7 of the protein samples. In the negative control, we can clearly see that while a sequence is detected, no insertions or deletions have been made to the sequence. In the negative result, it appears that a cut has been made. However, the sequence signal titled quality control shows a very skewed visual, showing that there was an error made in the sequencing of the sample, rendering its results suspicious and unusable. In addition, the indel spectrum shows that to the sides of the peaks and indels, there are no insertions or deletions, which also adds suspicion to the validity of the sample. The positive result, however, shows a relatively uniform sequence signal, and also appears to have an indel spectrum typical of sequences that are cut. To summarize the current findings of the project, the nanoparticles we constructed were shown to be successfully transfected in most of our cell samples. In addition, in some of our both mRNA and protein samples, a cut was successfully made in the fetal hemoglobin gene repressor. That being said, there is a lot of work to be done to further this project towards reaching its goals. First, we have to check the reproducibility of the results to ensure that the quality of the results and the conclusions made are not based around random chance. If the results are legitimately reproducible, it means that something biologically significant is occurring, which would affirm the hypotheses of the project. In addition, we have to check the titrate ratios further to increase the efficiency of the nanoparticles. We were unable to definitively judge which ratios proved most efficient, so further repetition of these nanoparticle experiments is needed. We would also like to transition over to using MiSeq to analyze the sequences, which will provide us more precise and detailed sequencing data, allowing us to analyze the results further. Finally, we have limited ourselves in this project to only target CD34 positive cells, which is unrealistic and inefficient from a clinical perspective. Once the results of this experiment have proved reproducible, we would like to move to using a broader scope of cells by targeting the treatment to CD90 positive cells. Overall, while much progress has been made towards the direction of successfully reactivating fetal hemoglobin in some cell types, there is much room for us to make the application of treatment broader and more efficient. Work that will hopefully continue over the next few months, granted we receive the proper funding to continue. Thank you for listening.